All right, here we're continuing with Chapter 9, Physical and Chemical Controls of Microbes. Remember, this is focused primarily outside of the body. And starting with Section 2 of Chapter 9, we have the remaining of the objectives to cover, except for Objective 11, which is your homework assignment. So you've got the 6 methods of physical control of microorganisms, which include heat, which is moist and dry. I'd like you to be able to define microbial growth and understand the use of cold and desiccation for microbial death or preservation, or just simply halting bacterial growth or metabolism. How is radiation used as a method of microbial control? And then understand filtration and osmotic pressure to an extent as control methods. All right, so this is where we left off for Part 1, Section 1, Part 2. And it's looking at the different targets for the physical and chemical agents: the cell wall, the cell membrane, cellular synthesis, and proteins. Realize that any one of those targets, if you disrupt one of them, it will eventually affect the entire cell. And so the bacterium, if that's what you're targeting, will eventually succumb to the environmental pressures and simply degrade, depending on how bad the action is. So if proteins are targeted, then the proteins are going to be stopped from being able to function. Cellular metabolism, cellular synthesis is not going to occur. The cell membrane will eventually lose its integrity, and the cell wall will eventually lose its integrity. All right, so first off, looking at the cell membrane, the major action here, or the major agent, is going to be surfactants. Surfactants are lipids, basically, all right? And those are going to be found in soaps, in detergents. What happens is that the surfactant molecule, because it's a lipid, actually looks and acts very similarly to the phospholipids in the plasma membrane, in the phospholipid bilayer here. And once enough of these surfactant molecules get pulled into the plasma membrane, the cell membrane, what happens is holes are created. Right? So then the cell membrane is no longer able to maintain integrity. Material that should stay out goes in. Material that should stay in goes out. And you simply lose the integrity of that cell, and it's going to rupture. Then, heat. Right? We all know that heat can kill. If you, unfortunately, were to leave a small child in a hot car, what happens? The child's body temperature increases, the enzymes will start to degrade, not work anymore, and that child could potentially die. So heat, in terms of elevated temperatures, will kill bacteria cell, the microorganism. Lower temperatures, on the other side, are microbistatic. All right, they're going to halt metabolism. Won't kill it, but it's just not going to work as fast and work as well. So if you think about you and going out for just to work or play a soccer game, watch a soccer game, if the temperature is perfect, 75 degrees, kind of sunny, you really feel good out there playing or watching. Drop the temperature down to 35 degrees, and you start moving slower. If you're sitting in the chair, you might start to kind of cramp up a little bit because you're cold and your muscles are trying to keep you warm, but it's not working very well. So the metabolism just simply slows down. When we're talking about heat, there's moist heat and dry heat. Moist heat is going to be hot water. It's going to be boiling water, steam. somewhere between the temperatures of 60 degrees Celsius and 135 degrees Celsius. And what's body temperature? 37 degrees Celsius. So even though 60 degrees being hot water may not seem like it would be very effective, it can be in some circumstances. Then there's dry heat, which is simply going to be hot air or an open flame. If you're in the lab, you should be using Bunsen burners to sterilize your inoculating loops, and that is an open flame. It's hot enough to burn off any material, any microorganisms, any contaminant off those wire loops. And so the ranges for dry heat can be 160 degrees Celsius to thousands of degrees. And that's when you're going to see incineration effects. All right, when you're looking at heat, an important thing to remember is time, because you can't just put something at 60 degrees for 2 minutes and expect it to do its job. Whereas if you put your inoculating loop into your flame for 2 minutes, it's going to be quite crispy. All right, there's nothing viable left on that piece of metal. All right, so just a different way to look at moist heat. You have boiling, you have autoclaving, and pasteurization. Boiling is boiling. All right, we boil water on the stove, in the microwave, and what that does is it kills vegetative cells, so the metabolically active cells, bacteria, fungi, and then most viruses. And as I mentioned on the previous slide, time is important. All right, again, just bringing something to a boil may not work 100%, especially organisms like fungi that have a rather sophisticated cell wall, or some of them can have a sophisticated cell wall. Then there's autoclaving, which is going to combine high pressure and high temperature, and there you're going to see sterilization. And sterilization, if you remember, is the death of all microorganisms, including bacterial endospores. And autoclaving is used a lot in the microbiology lab because it's easy to sterilize media, and then your cultures, your material to inoculate to perform the different tests that you want to look at are free of contaminating microorganisms. So then you can say, E. coli did this, and not have to worry, was that a fungal contaminant from the air? Was that staph or something like that? And then pasteurization is also moist heat, and it's used primarily for food purposes. Milk, ice cream, yogurt, fruit juices, to make that food material safe. Because yes, milk, fruit juices especially, those actually can contain microorganisms just through the process of making them. All right, the fruit, your apple on the fruit tree, is not sterile. And so what pasteurization does is it just simply kills off the organisms that don't like the heat. Right? So organisms that are going to be pathogenic to humans. But the heat tolerant, the heat loving organisms are able to survive, which in reality that is fine, because if they like the heat, our bodies are not hot. All right? Heat tolerant, in terms of pasteurization, the temperature for pasteurization is usually around 70 degrees. Time is important, so you can bring it up a little higher, but then you just decrease the time. And so you don't kill off those really, got the guys who really like the high temperature, but those aren't going to cause problems, so it's not a big deal. All right, then with cold and desiccation. All right, principal benefit of cold treatment is just to slow the growth of the cultures. And with the cold, a lot of times we're talking about our food. So we refrigerate food so that any contaminating microbes are going to grow a lot slower. You can put food in the refrigerator, and over time, it can go bad like milk. You can have cheese that is a little furry after a while of not using it. You can have other foods, fruit, vegetables. I've even seen mold in jelly jars and in salsa containers. All right, so over time, those organisms can still grow, but they're not going to grow very fast. And so if you put something in the refrigerator and you eat it after 3 days, you're going to be safe. So the cold merely retards the metabolic activities of most microbes. And usually, if the process is done right, microbes are not adversely affected by gradual cooling or long-term refrigeration or deep freezing. All right, so that's good because for the microbiology lab and for scientists, we can store samples and then be able to come back to them, take a sample out of the refrigerator or out of the freezer and grow that up and use it again for student experiments. All right, and a lot of times those temperatures are very cold with that deep freezing, minus 70 degrees Celsius to minus 135 degrees Celsius. That's pretty cold. And you can keep bacteria cultures alive, but frozen, for decades. Viruses and fungi can be kept for decades. And then thawed out, put in the right medium, and they can grow and survive and thrive. All right, radiation. This can be a debatable topic in terms of whether or not people like radiation as a means of microbial growth. But what radiation does is it messes up our DNA. And here you have the normal segment of DNA where these two thymines are connecting through the hydrogen bonds across from each other with the adenines. Whereas with the thymine dimers, what happens is it causes the radiation exposure causes these two thymines to develop bonds between the two of them. And that messes up the machinery, the cellular machinery of the cell when this piece of DNA goes to be replicated. And so once it gets to this spot here with the thymine dimers, the machinery doesn't know how to read that. And so it can often just add random nucleotides into it. And as we learned from Chapter 8, random nucleotides could end up leading to mutations. Mutations could lead to funky proteins, inactive proteins, oncogenes, those type of things. So we want to avoid radiation to our cells, but we can use it to do this to bacteria cells and cause death there. And this is a before and after, basically. Before radiation, non-irradiated strawberries, you can see how slimy they are, how moldy. And these are the irradiated, the treated strawberries, free of the microbes. Some people will argue that irradiating our food is going to alter the taste. My non-educated, so to speak, opinion of that is that it's not going to. The UV radiation only penetrates what's on the surface. And so the microbes, the fungal organisms on the surface, if it's only put under the UV light exposure for a short amount of time, it's going to kill the fungal organisms. And then if pulled out of the UV radiation, it's not going to penetrate the strawberry cells for long enough to actually alter the taste. All right, here is filtration, where you have a filter that has different size pores, and you strain liquid or air, and all the microbes stay on this side, and the material underneath that has passed through that filter is going to be sterile. And liquids and air are the major things that are going to be filtered. Obviously, solids are not. All right, and this is just an introduction to Section 3, which you have a YouTube video to watch because that is already a very good lecture, so to speak, of that material.